Hello everyone, I'm Priyanka and today we will talk about colorimetry. The topic will be covered under the following headings: introduction, principle, laws, flow representation, instrumentation, and application. Firstly, let us talk about electromagnetic spectrum. Electromagnetic spectrum is the full range of wavelength of light. Electromagnetic spectrum includes very low frequency radio waves, microwaves, infrared, visible, and ultraviolet light to X-ray and gamma rays. As you can see in the figure, uh, short length, short wavelength, high frequency, high energy. Short wavelength, shorter the wavelength, higher will be the frequency, and higher will be the frequency, that means higher energy is there. Moving to the next, and lower frequency, lower wavelength, and lower will be the energy. As in our previous study, we have learned about the light is a type of electromagnetic radiation consists of different wavelengths which can be decomposed by prism, produces a color spectrum. Now think about what is wavelength. Uh, Wavelength is the length of the light wave which determines its color, commonly designated by a Greek letter lambda. As you can see here, wavelength is the distance from this crust to the another peak, another crust level. Okay, so a wave have a crust and a trough. The distance between two crusts is your wavelength. Wavelength measured by the unit uh, nanometers, micron. So the colorimeter is an instrument which is used in the measurement of luminescence intensity of light. So what happens? In colorimeter, first of all the sample must be a colored solution, and then the colorimeter will measure the intensity of the light absorbed by the sample, that is the absorbance. Colorimeter involves the quantitative estimation of colors. The difference in the color intensity result in the difference in the absorption of light. The intensity of the color is directly proportional to the concentration of the compound being measured. The amount of the light absorbed or transmitted by a color solution depend on two principles, that is Beer's law and Lambert's law. Beer's law states the absorbance is directly proportional to the concentration, whereas the Lambert law says absorbance is directly proportional to the path length. Next we have derivation of the formula. Combining the two laws, absorbance is directly proportional to concentration into L, that is path length. Or we can write A is equal to K, that is the constant here, into C, that is the concentration, into the L, that is path length. Let AT, that is the absorbance of the test solution, CT, that is concentration of test solution, AS, that is absorbance of standard solution, CS, that is concentration of standard solution. So we have equation AT is equal to K into CT into L. This one for the test and another one for the standard. AS is equal to K into CS into L. Placing the equations here as A T upon AS is equal to K into CT into L upon K into CS into L. Simplifying the equation, both the constant are removed and the length also. So we remain with AT upon S is equal to CT upon CS. So taking a CT at one side, we have AT upon AS into CS. By using this formula, we can estimate the concentration of the test in the sample. So we will place the absorbance of the test divided by the absorbance of standard into the concentration of the standard. So next we have CT is equal to AT upon AS into the CS. So the concentration of the test solution can be estimated by this formula. Absorbance of test upon absorbance of standard into the concentration of the standard. So this is the standard formula by which we can estimate the concentration of the test solution. In case we want to measure the concentration of the test solution per 100 ml, the formula will be absorbance of the test divided by the absorbance of the standard into the concentration of the standard into 100 upon x ml, like whatever the quantity you want to measure. So next, we simplify the formula by concentration of the test upon 100 ml is equal to absorbance of the test divided by the absorbance of standard into the concentration of the standard into 100 upon x ml. So next we will simplify this formula. Concentration of the test upon per 100 ml is equal to OD, that is absorbance of test, minus OD of B. B here is the blank solution. OD upon the OD of S minus the OD of the B, like blank solution, into the absorbance of into amount of S divided by the volume of the T into 100. So the concentration of the test per 100 ml is equal to T minus P upon S minus P into the amount of S into 100 divided by the volume of the T. Next we have the flow representation of colorimeter. First we have a light source, visible light source. Next we have the slits, mirrors, lenses and slits. After that, a filter or a diffraction grading is placed. Then we place our color solution and we have a photocell. Now this photocell will convert the light energy into the electrical energy. Then we have a digital output of absorbance. So as you can see the diagrammatic representation, first we have the light source, then we have slit to obtain a parallel rays. Okay, when we have a lens system, then our color glass filter, then we have a cell for the sample, then photoelectric cell, then a galvanometer is attached to it. Talking about the instrumentation part of the colorimeter, first one is the light source, then slits, then condensing lens, filter, detector, that is a photocell, and then a output. Contemporary filters for colored solution. The selected filter has the color to the contemporary to that of the color of the unknown solution. First thing that I have mentioned, that the solution must be a colored solution. In colorimeter, according to the color of the solution, we select the filter to be used. Like if we have a yellow color solution, then we use blue color filter. This blue color filter have the wavelength ranging from 450 nanometer to 479 nanometer. If our color solution is red, then we use green filter. Now this green filter having the wavelength around uh, 5052 to 537 nanometer. If the solution is blue, then we use a yellow filter having the wavelength ranging from 640 to 689. If our solution is green, then we use a red color filter which have wavelength ranging from 620 to 689. We place our sample in cuvettes. These cuvettes are simple sample holders. Cuvettes are rectangular cells, square cells, or circular ones made up of optical glass for visible wavelength. Common one is square rectangular to avoid refraction artifacts. Dimension of cuvette is 1 centimeter. One side of the cuvette is made up of quartz and another is made up of glass. When we place our sample in a spectrophotometer or colorimeter, we prefer the glass side to be placed towards the light source. As you can see here, one clear side is there and one somewhat blur side. This blur side is made up of quartz and another side is made up of glass. Light beam will enter from the glass side. Next we have application of colorimetric assay. For the estimation of biochemical samples like plasma, serum, CSF, urine. For example, determination of blood glucose, blood urea, serum creatinine, serum protein, serum cholesterol, serum inorganic phosphate, serum urine creatinine level, and glucose in CSF, etc. They are used by the food industry and by manufacturers of paints and textiles. Thank you.